Hello everyone, this video is about Propidium iodide or PI staining. So this Propidium iodide is a red fluorescent dye. This can intercalate between base pairs of DNA. So make sure you are using gloves when handling this chemical, and this can be used to stain cells and nucleic acids. So you can take a look at dead cells or count the number of cells because these can bind to the DNA in the nucleus. To detect cells, the theory is after getting damaged the cell membranes by reactive oxygen species, this dye can penetrate to these damaged cells and go bind to the DNA, so you can visualize these dead cells. And some characters, this has a molecular weight around 666.8 moles per gram, and you can store this chemical 2 to 6 Celsius, and make sure you are protecting this from light. And this is soluble in water or any buffers. And there are different published methods. You can use any of them. So the method I'm using is, first I dissolve this chemical as 5 to 10 micrograms or 1 grams per ml in distilled water or PBS. And then I incubate these seedlings 2 to 5 minutes under dark conditions. Make sure you are not keeping this too long because it can overstain your samples. And then after washing 3 times with the distilled water or buffer, you can visualize these under the fluorescence microscope and use the RFP filters. And you can obtain 2 things from this. You can visualize the dead cells under stress conditions as seen in these Arabidopsis reeds. This is how dead regions are visualized under the fluorescence microscope. And also you can count cells. As you can see, the nucleus are stained. So you can count these cells using this staining procedure. And good luck!